then I need to say that we are still being live streamed and recorded through the Q&A session. So I don't have much in the way of announcements. I can pull up my slides from before if we need to in a minute. I guess since this is technically the last day of the workshop, there's not much to announce for the next day of the workshop because now we're in the hackathon. So I do just want to put up our list of people to contact again, in case you need to get in touch with anybody directly from any point in the workshop. The one thing I did want to say is that we will have a exit survey about your experience with the overall workshop. We'll be sending that out by email later tonight. So please do take a couple of minutes here or when you get a chance to fill that out for us. It's very helpful in how we structure next year's workshop and if there's anything that you feel would be useful to include for next year. So again, that'll be going to your emails later tonight. So with that, I figure we'll kick off the Q&A discussion. 3 more minutes on the poll. So this session, I figured we could be a little bit more informal if people would like to unmute and ask questions about the hackathon projects that they've got coming up. We are ready to help answer questions or help people organize. I guess what do you want to say the next?
Thanks to everybody. We'll just say thank you to everybody who stuck around till the end of this. This is a long, long week. Of course, we still have 2 more days.
Yeah, I guess the people that are still here are going to be sticking around for 2 more days, but yeah, we do appreciate it. It was really nice this year to see so many of the same people showing up every day. So consistently too. There's very little drop off, which we're glad to see. Hopefully that means that the workshop was very useful for you.
Yeah, sorry. So I think during our discussion on the hackathon in the afternoon, you said students who wanted to work alone, and then asking for maybe some kind of assistance. You are planning on creating a separate room for them. Can you elaborate on that?
Yeah, so that was something that came up during the afternoon today, was that we had a couple of people who would prefer to work alone on their project and a couple of people who are having difficulty finding others interested in their project. And we thought that it might be useful to have, so we're going to have breakout rooms for each project tomorrow, but we thought it might be useful to also have a kind of like independent work breakout room where people working on their own projects can sit while they work, and if you have questions, you can relay those questions to the others working independently, because you may be having some of the same issues across projects. Instead of just having you working totally by yourself, and then if you do need help from an instructor, we can send somebody over. Does that kind of help clarify what we meant by that?
We'll create a bunch of extra breakout rooms that people could use. If people want to join, have their own and just sit there, that's fine. If people would like to sit together in a virtual room, that's fine too.
Yeah, so maybe that's actually a good way to do it, is we can have people who are working independently just sit in the main room during the hackathon, and we'll have a couple of extra breakout rooms that you can move to if you'd like to discuss something with someone who is also working independently in more detail.
Okay. Yeah, thank you.
Any other hackathon questions? Happy to answer questions about format as well. Doesn't just have to be teaming.
Or questions for Julio.
Yeah, more questions
or
for
TJ.
Julia.
Gilberto is here. I'm here.
I do notice James is here, James Duran.
Yes.
Or Duran. You guys had something specific to talk about, I think, at some point. So Sharon, I also noticed you unmuted if you'd like to go ahead.
Well, I had something something for TJ.
Okay.
Is he here?
Yes, I'm here, Sharon. Go ahead.
You may remember earlier in the workshop, I had an issue with the randomness. It seemed like the seed wasn't being randomized because I would run a random simulation and it was the same each time. I have now, and I sent you my code. I just today in the parallel session experienced something similar, where we were using to divide the cell in the random direction, and mine always divided in the same direction, which is odd. But I have a video and I can send you the code as well.
Yeah, that would be very helpful. You're on a Mac, correct?
I'm on a Mac. Is this something that might be platform specific?
It's highly unlikely, but it's hard for me to imagine any other difference because I've not seen this behavior on Windows or Linux. But,
Well, it will be interesting to touch base with you later and see if we figure out what the cause is.
Yep. Yeah, thank you for bringing that to our attention and for documenting it.
Okay.
And it seems very quiet, so if there are no questions, you certainly don't have to stick around. I know poor James has been waiting a long time to talk to me. If you want to do that not in public, we can find a place to do it too.
I'm happy to talk wherever Dr. Cozy is.
Otherwise, just here is fine. So
yeah, that works fine for me. Yeah, I saw you offering to help get the team sorted earlier. I appreciate that. Sorry, I missed the message at the time I was having dinner, but yeah, we still have 3 at this point, so just need just a couple more people. I think the person I was saying about the difficulties, Jacob, I'm not sure how you pronounce it. I think he's with us now, so if he still has any issues, I guess could be the chance to discuss them.
So Jakob, did you say you were having trouble getting CompuCell to run on your computer?
Yes, I actually did. And if I could get any one of you to a breakout room, I would be very grateful.
That would be great. Who would like to help Jakob?
What's the issue, Jakob? You're having
What's
nanohub
happening?
crashes, right?
I mean, what I actually managed to solve most of my troubles by just setting up Linux on my second laptop and configuring CompuCell on Linux, and that magically worked.
That might be the first time I've ever heard somebody of solving their problems by setting up CompuCell on Linux, but
yeah, that actually worked. So
okay, well, yeah, if you still want to talk to somebody, maybe Joel, if you're here, or Pedro might be able to help you in a breakout room.
yeah, I still have some minor issues, and it would be good to discuss them before the hackathon session.
Okay. Joel, do you have a minute, or would you prefer if Pedro took this one?
Sure, I can take a look.
Okay, great. Yeah, so if you guys just want to move to, I don't know if Joel has his own breakout room, but you can, yeah, he does, okay.
All right, thank you.
Sarana asks, I am interested in thinking through how to scale from hundreds of thousands of cells. Could you elaborate on that a little bit,
I can
Sarana?
elaborate more. So
Sure.
what I'm planning to do is looking at like cell transitioning, like some primordial germ cells to like the transitory, and looking at like clustering. Basically, I'm interested in modeling how they cluster and germ cell lines. And well, it's based on like the density of the number of primordial follicle cells. And for females, that's like 500,000. And so some of like other computational models that I've used, we can easily just put in like 300, 400, 500,000, like just simulating monocardal simulations, but I'm not quite sure if it's feasible because I understand it will take a long time if I were to put that many, specify that many cells, but that's also something that I can't just put maybe one cell because it's really, well, I mean, I can start to parameterize something with one cell, but really trying to get it with some practical implications, I'm going to have to get several thousands of cells in there.
Well, so several thousand cells is no problem.
Yeah, but like, I again, so that's why
10,000 cells is no problem.
I'm not quite sure how much to. I know that you had given a couple of suggestions, and which I have noted in my plan of action for tomorrow,
Right.
but typically on the conservative side, I still use 300,000 on like,
So
again,
what modeling methods are you using now? What package are you using?
well, so I haven't done any like virtual tissue modeling. So before I was using MATLAB, I've done Berkeley Madonna. Those
So these are agent-based models or these are network models?
agent-based model. Oh, actually, so the one I did in MATLAB was definitely network-based. And so I've done both essentially.
I mean, usually the issue is the level of spatial detail that you need. If you need a lot of spatial detail, then it costs more per cell. If you could get away with cellular automata, then doing 1 million cells is no problem. But then the cells don't move. They don't rearrange. If you use center models, then 100,000 cells is relatively easy.
You say
But
by
the cells-
center, you say
center
center
models, yes,
Oh.
molecular dynamics style models. But then the cells have no shape. And so the question usually is whether the cell shape is important. If it is, then the kinds of modeling that we do here are useful, but you pay a price because the extra detail that you have in each cell takes more computer time.
Yeah, and so the details are important, but I'm just interested in a few very specific like key event details because of what has been supported, of course, in the lab and what has been like in vivo studies and in human studies. And so there's not that much, honestly, that's already out there. So I'm also for this particular topic. And so I'm open to exploring things that could just make sense because of how complicated it is. There's just very little modeling of all of this.
Right. Well, I think maybe then we should tomorrow look at some of the models you've done and ask the question, what does it take to translate those into the CompuCell framework?
Okay.
And what additional benefit would you have by doing that? Again, the point is that usually when you're dealing with tissues, I mean, there's some tissues where you really need to model the whole tissue, but most tissues have a functional unit which doesn't have that many cells in it. So if you're dealing with the liver, you don't model a whole liver, you model a sinusoid rather than the whole lobule. If you're doing a kidney, you model one tubule, you don't model the entire kidney.
Right.
If you're modeling the heart, you might model the whole heart, but not include cells. But you might model a patch of a few millimeters by a few millimeters of the heart, where you include the biofibers explicitly. And so I think the question is whether you need to, usually you don't need to model all the cells over square cubic centimeters at the same time. And people tend to do a continuum model of the whole tissue, and then couple that to models of the individual cells in small volumes. So those are the kinds of things that can be thought about.
Yeah, I've been thinking about like ideally I wanted to do all of them because like looking at the ovary, the more specific thing within the ovary is the follicle, and a lot of the behaviors with the quiescent follicles is based on the density, and it's just like the more
Right. So
that they're
then you
clustered
pick
together.
a patch of 100 of them where you can get the density. You don't do the whole ovary at once. And so I think typically that the question is if you could define a subvolume which has enough structure that it gives you the detailed architecture. And then you think of that as being repeated many, many times. It's
Okay, yeah,
usually-
I'll have that. I definitely have that because I've thought of also about like a sub, but that's why I haven't quite.
Right.
Again, I just been looking at all of it, like
Right.
thinking about, okay, try to see what makes sense.
Right.
But
But for example, if there's a particular cell type that's very important, you might represent individual ones. But then you have things like supporting cells that may not be moving around or doing very much except providing a growth factor or providing a substrate. And then you might coarse grain those and present those as a block of tissue without modeling individual cells. I mean, there are definitely tissue structures where you need to model a lot of cells to see what's going on. I'm not saying that, but in many cases, you can the actual number of cells that are actually doing something that you'd care about is few. There was a sort of philosophical. I think it's relevant for the hackathon ideas, especially given some of the things I was hearing the other day. We had this nice talk a year ago by Rusty Irving, who was the inventor of the digital twin concept at General Electric. And he talked about how he worked with the military a lot, building digital twins. And he would go and he would say, there'd be some admiral. The admiral would say, I want you to build a digital twin of a battleship. Actually, in this case, an aircraft carrier. And they'd say, I want you to model everything in the aircraft carrier. I want you to model every motor in the aircraft carrier, every plate of steel, every gun, every turret. And Rusty would say, well, we could do that, but it'll cost you $10 billion. It'll take 20 years to build the model. And he said, what do you need it for? And the admiral says, well, the elevators that are taking the planes up to the deck are jamming. And Rusty said, well, what if we just model the elevator? And the response was, I want a model of everything. And the response was, okay. But the point was that if you can define some subcomponents that you can model more easily, then you're much more likely to succeed. And in the end, Rusty did convince the admiral that they should just model the elevator, and it worked. Now, it's not always possible. Maybe the elevator is failing because somebody in the kitchen is doing something, and it's not included in the model. But to begin with, I think it's important to try to focus on the things that you think are the most important in a local environment. Because one thing we know is that there is no long-range information in tissues. It's all local information. Cells don't know what's happening 20 cell diameters away. They only know what's happening locally. And so, as a matter of principle of biological organization, unless you're talking about transport in the blood or the lymph, information is local, and that means tissue organization has to be maintained locally. Now, it may be 10 cell diameters, 20 cell diameters. 20 cells by 20 cells by 20 cells is still 10,000s of cells. But it's not 10 billion cells. Now, in the case, I know people like Cassandra Expervor, who works in OSI development, and she does in insects. You get these very complicated, large-scale structures in the ovaries, and in that case, if you want to model the whole thing, you need a lot of cells. So I think it's important to look carefully at what the fundamental structure is that you're looking at, and ask the question, at least to begin with, what's the simplest model you could build? And clearly, if you built elaborate models that work, you want to start with those and ask the question how you could use spatial modeling and agents to improve them. Jacob, were you able to get your problem debugged, or do you need to talk to someone else? Or?
I still need some help because we were not able to solve it completely.
Well, you're using Linux. As we said at the beginning, we won't help people with Linux. We can't. What kind of computer?
And
do
the
you
same
have?
issue on Windows.
No, that's not possible. Genuinely not possible.
Not the chance.
So if it's Windows, TJ can get you fixed.
I could probably help with Linux too.
Okay, so why don't you two go and bash it out?
Yeah, Jacob, let's head to my breakout room.
All right.
So yeah, I'll probably need someone else to help us there. I did everything I know, and then my computer works, unfortunately, on his computer. That's the worst type of thing that we can say, right? It runs on my machine.
But I genuinely don't believe that there's a fundamental problem with CompuCell on a Windows console.
No, yeah, because
that's not happening. But Macs, yes. Linux, yes. NanoHub, yes. Which is like NanoHub is Linux. It's part of the
right.
But Windows, no.
Yeah, yeah. Did you assign him to someone else?
I think TJ is.
Yeah, TJ is the best. Yeah. Okay.
So James, did you want to talk? We've been waiting patiently. It's midnight for you, so why don't we talk about what you want to do?
Yeah, of course.
You're welcome to take the screen share if you like. You need to.
Sure.
Sorry, let me grab the screen share back. And I'll yeah, it's yours.
Yeah, so I think sort of a rough plan of action is, I think initially just try and get the basics of the initial model nailed down. So I think, try and keep limited cell populations. Perhaps maybe not cytokine start with just like a generic chemokine, and just try and get the original some of the original results from this paper. There were a few areas for future work they suggested that I guess once that model's working, we could then look into. I think, for example, the original paper had no flux boundary conditions, and there was a feeling that that might be impacting on the ability for granuloma to form, because sort of near the boundaries or near the corners, things were getting compressed in, and whereas with periodic boundary conditions, there might be different outcomes there. And then I thought, if we're able to get all that done, it might be overly ambitious, I'm not sure, but I remember you saying about the papers that Denise Kirshner's group wrote, and I know the first one, or one of the first they did, was like an ODE model with a lot of cytokines and differentiated T cells and stuff. And so maybe trying to convert that into CompuCell, perhaps using that paper that TJ and a few of you wrote on the cellularization of ODEs into agent-based models could be a way of trying to just make a more complex agent-based model of the within-host TB dynamics. So I think I'm not sure how much more time that would take, obviously, because then it's converting the parameters through that process as well. So I don't know if that's overly ambitious or not, but I think as a rough aim for the 2 days, that was kind of what I was thinking as an initial plan.
Well, so I think it's a little bit like what Sarana was talking about, which is the same issue, which is what level of complexity do you want to put in when? And so with granuloma, I think there are two fundamental questions, one of which is what's the spatial organization of the granuloma? How do you get the bacteria surrounded by perhaps dying or some kind of non-cellular material surrounded by some early immune cells surrounded by stromal tissues? And then are you going to model that in 2 dimensions or 3 dimensions? It's much easier to model it in 2D, but of course, sometimes 3D is very important. And if you could build that spatial organization, then that is replicate her results or other results of the spatial organization. That's already quite interesting. The other question is, once you can replicate that spatial organization, perhaps with some simple signaling where you have a couple of cytokines or chemokines that are pulling your immune cells in, the cells are doing the right thing, they're dying, they're killing it, they're eating the bacteria replicating and dying at a slow rate, and so on. Then you could ask the question, now what if we put in a more detailed biochemical model? And now we can ask the question, how does disruption happen? If we hit the system with a steroid, what happens? If we hit the system with a pro-inflammatory signal, what happens? And so I think you could really break the problem down to 2 parts, one of which is building something that gives you the spatial architecture, and one of which is how do you get the detailed feedback loops in the immune signals that are responsible for creating and maintaining and disrupting the integrity of the granuloma. As you point out, Denise was able to do the feedback models without including the spatial component. She certainly built plenty of models with the spatial component. And so I wouldn't tell you which way to go, but I might first, my suggestion is that probably getting the spatial things done is not that hard. So making sure that you can get the spatial organization to be more or less appropriate is probably a good start. And then if there are existing models of the biochemical networks that work, importing those is not that hard. And so, now what that paper of TJ's about cellularization is trying to do is to point out that if you're careful, you don't have to reparameterize things. That if the parameters exist in an ODE model and they're validated, you can pull them in without having to tune them up by hand. So that's already very, very valuable. But that probably won't buy you the spatial model. But I know Denise has spatial models. And so trying to put those 2 things together, I think could be very interesting. I think it's, as I say, it's something I've wanted to see somebody do for a long time. I've looked at Denise's models for years and thought those should be really easy to replicate. I'm sure they're hard. I'm sure they're harder than they look. But I don't see anything in those models that's intrinsically not available in CompuCell. The scale of the models is reasonable. The one thing that you don't have is she doesn't really publish her methods. And so you may have to guess some of the parameters and some of the techniques that she uses.
Right. Yeah.
But, I think it's practical. So I would pick 1 or 2 papers to replicate to begin with, and see how far you get. But I think it should be reasonable. The idea of having a cluster of bacteria, which are sending out a chemo-attracted signal, surrounded by immune cells, neutrophils, resident macrophages, and so on, some sort of non-cellular debris produced by that should be realizable. And then if you have the networks, layering them in isn't that hard. So I think that's a great project, and I hope you could attract some people to do it, because it'll be a great learning experience. It's got a good, it's an ideal, almost ideal level of difficulty for a 2-day hack.
Yeah, thanks for all the advice. So it sounds really useful thing to pick up. So yeah, thanks.
Well, I don't know what to do in the sense that I wish I could recruit, crack a whip and recruit people for you. If I were good, I mean, they're all good projects, but I have to say, if I were going to pick one, and unfortunately, because as an organizer, I don't get to just join a group. But if Lorenzo winds up not being part of a group, or his own group, for example, you might be able to pull him in because he's done a lot of work on spatial organization at CompuCell and in layering in complex network models. And so he has a lot of expertise. He's been working in vascular development, but he definitely has the knowledge to implement a model of this
kind. Oh, perfect. Yeah. If he wants to join, then yeah, I'd certainly be very happy to have him.
So I might send an email to him directly about that. I think
yeah, he said today that he may just focus on helping out during the hackathon instead of working on his idea. So if you could probably recruit him to give you guys a good bit of help.
Perfect.
And he's in your time zone.
Ah, even better.
In London. So
great.
So is at King's College. So he's somebody that's not too far away.
No, definitely not. No. Perfect. Thank you.
And he's very interested in infectious disease. He's been one of the co-leads of our working group on infectious disease and immune response.
Oh, all right.
So even though his main work is in vascular biology, he has a strong interest in this. He certainly heard Denise's presentation. He'll know about granulomas.
Excellent. Yeah, sounds good. I'll have to get in touch.
I don't know if anybody else in the room has any ideas about that, but I think, and I'll come in and help as much as I can. As I say, I wind up having to go around the room, but I'll be glad to come in and do some brainstorming and troubleshooting, put up the papers, help design the model.
Perfect. That sounds great. Thank you. Cheers.
Great. Get some rest, please. It's going to be a long 2 days. Long 2 days now, so please get some something to eat and go to get some rest. And I know.
Yeah, that sounds good. It's almost in my head. So, yeah.
The time zones, as I say, may have abolished distance, but we haven't abolished time zone. Although given queues in English airports, I think it's probably just as well not to be trying to go in person.
True.
Okay. Is there anything else, or is that I'm sorry not to give you a definitive answer, but I think your strategy seems reasonable.
No, that's all been very helpful. So no, that all sounds great. Thank you. So, yeah.
Okay, great. Okay.
Right.
Good night then. Okay. And Sarana, I don't want to be discouraging to you at all. I just am trying to help you narrow down something that's the first achievable step. And so my suggestion again would be to pick a small volume of tissue, simulate a tiny volume of tissue, and then build out. That would be how. Very much what I was saying with James. Start with a small thing and build out.
Yeah, that'll help you a lot in the prototyping phase too, just because it'll run faster.
Sarah, did you see the presentation of Shane? So Shane basically he simulated what would be a big tissue, a small version of it, and still got very impressive results from it. So I think you can take an approach like him.
I don't know if she's on the call or not.
But I think chain simulation is a perfect example of this.
Right, because the palette is really very large and it's
Yes.
three-dimensional. And if you tried to simulate it in full 3D, you couldn't do it. You'd have to do a continuum model. And if you did a continuum model, you'd lose all of that biochemical signaling and the structure of how the cells are rearranged. And so doing that sagittal section, I think, has worked really well. Any other questions, comments?
I have a totally irrelevant comment.
It's okay. That's
But
fine.
the concept of distance is fundamental in physics and in math even more so. And it was used in some presentations during the week. And my wonder is, very far from CC3D probably, but maybe it can be checked there. Maybe some results or patterns may vary as a function of what definition of distance we're using. And in networks and graphs, distance is, there are so many variant and definitions of distances. Anyway.
Well, I
I'm
think
not
that's true. I think that's true. And there's a problem with distance in CompuCell, which is that we're on a lattice. So distances aren't Euclidean.
Right.
A lot of the time that
3D,
doesn't matter.
in 3D,
Doesn't
they
matter.
can be, in 3D, they can be geodesic.
Another issue that comes up another issue that could come up is dimensionality. A lot of times you can get away with a 2D model. If you're in an epithelium, you look from the top away, and you see basically the surface of the cells. But you also are losing information. If the cells appear to contract, it's not that their volume is changing, it's that they're becoming more columnar and less squamous. So you don't see volume conservation anymore when you make a 2D version of a 3D epithelium. In the same way, if you do sagittal section and you do something like clustering, the apical or basal domains, depending on the situation, getting rosettes, that works. But there are things that happen in 3D that don't happen in 2D. Famously, if you have a soap bubble in 3D, you make a hole in it, the hole closes because of the line tension around the hole. In 2D, if you make a hole, the bubble disappears because there's no information that goes between the edges of the hole, because that goes in the third dimension to get that information. And so there are certainly times when in 2D, if I do a section, I can't tell if I'm in a spherical cyst or if I'm in a tube, because they look the same in section. But functionally, they're very different. And so there are certainly cases where 3D really matters, and the 2D approximations don't quite give you what you want. I gave the example of doing sinusoid for the liver. In a lot of tissues, the vasculature is more or less isotropic, and doing a 2D version is not so bad. Or looking at a single vessel in 2D isn't so bad. But in the liver, section of the liver lobule looks a little bit like a grapefruit. There's a radial organization of blood vessels. The density of the blood vessels changes depending on where you are radially. The gene expression changes depending on where you are radially. And so to assume that you can model a single sinusoid is a pretty harsh assumption. Because in fact, the situation at the portal triad, the outer end of the sinusoid, and the situation central vein in the middle are very, very different. Flow rates are different. The environment is very different. And so in that case, you probably at least need to do a 2D model of the grapefruit slice. And in fact, even in 3D, the 3D is important too, because you have a lot of cross coupling. And so there are times when the 3D structure, somebody was talking about modeling podocytes. Trying to model podocytes in 2D is, trying to model them in 3D is terrifying. Trying to model them in 2D is probably not going to work, depending on what you're trying to do. And so you always have to ask these questions about what complexity can you neglect and still keep the things you care about, and when is the complexity irreducible? Sometimes you're unlucky. If you want to build meshes to try to understand extracellular matrix, fundamentally in 2D, if you have two lines in a plane and you want to get from here to here and here to here, they cross. In 3D, they can go around each other. And so you have fundamental differences in the way rigidity transitions happen in 2D and 3D, for example. The fact that 3D simulations of fibers is incredibly expensive is just unfortunate. The 2D simulations don't give you the right answer. Actually, Giulio's an expert. He'll show you. He can tell you when the 2D simulations work and when they don't. So that's something he's worked on his work. I think you've raised some very fundamental questions.
Sounds like amazing. I thought that always there were these nonlinear and at least 3 dimensions, but I may be wrong in terms of practicality.
Oh, well, so if
linearization is the first pragmatic step.
Well, so they're different. They're a little bit different. So linearization of things like ODEs, absolutely, feedback is important, and assuming linear models and getting rid of feedback loops destroys things. If you're talking about the spatial equivalency of networks, right? Biochemical networks are often called hairballs. And the reason they're called hairballs is because there is no way to embed them in 2-dimensional or 3-dimensional space that makes sense. Because a network doesn't have to respect Euclidean geometry. And so you can't draw them effectively. So I think that's a good point. I mean, trying to actually draw representations of biochemical hairballs in 2D or 3D is tough. Cluster analysis, of course, they have these beautiful ways now of displaying things like flow cytometry data or genomics data, high dimensional genomics data, reducing it using principal component analysis. But fundamentally, a lot of these things, there really isn't a low dimensional representation that makes sense.